so I know that we're all awake. And I know we have competition because lunch is going on as well, so thank you all for being here. So let me try again. Good afternoon.
Hey.
Now I feel it. And I think that's very important when we're discussing such important things that we're all aware. Oh, thank you for the microphone. Aware. Can you hear me now? Better? Oh, good. Good to see you. So, thank you for joining us. My name is David Patrician. I am an American journalist based in Hamburg. I work for German media. It is a pleasure to be back here at the Falling Walls Science Summit for our discussion today, Neurodegeneration, Breakthroughs, and Novel Therapies. Now, before we begin, two things. One, just a quick historical note, away from the science for a moment. Today is November 9th. We are in Berlin. 36 years ago, the Berlin Wall fell. It is part of the naming of this summit. So I think it truly is amazing to be in this city on such a historic day. And for those that are visiting, I hope you have a chance just to go around a little bit and see some of the historic sites, because it really is amazing that 36 years later, we are here in old capital of East Germany in Berlin having this Falling Wall Science Summit. So that's a historical note. Second point, when we had our last discussion a few days ago, I was privileged to also lead the discussion about Alzheimer's. I wanted to ask in this room, who here are specialists when it comes to neurodegeneration? If you can put your hands up. Okay, so I'd say about maybe 20 to 30%. The reason I ask is that my job is to make sure that the few experts that are here, that they get something from our panel, and they are all extremely excellent, but also those that are kind of new to this session or new to this subject can also take away a lot from the exciting studies that are going on. So with that, just a quick background. As of recent estimates, in 2023, around 56 million people worldwide are living with dementia. And Alzheimer's disease accounts for about 60% of these cases. This means that unfortunately around 35 million people globally are affected by Alzheimer's. As life expectancy rises, that's a good thing, but on the other side, so do the numbers of patients with Alzheimer's. We expect a doubling of the number of people with neurodegenerative diseases within the next 30 years. It's truly a startling fact. So the race to slow down these diseases, as well as other neurodegenerative disorders, is certainly on. Where are we now regarding or looking for cures? And can we slow down and finally cure its advancements? We're going to talk about this. And before I begin, I want to just go through with a raise of hands, again, interactive. If you can tell me, do you know someone who unfortunately is suffering from Alzheimer's disease? Put your hands up, just so we get a feel for it. So pretty much the entire room. Alzheimer's affects memory and cognitive function. Just for comparison, Parkinson's disease is also one. Do you know of anyone here in the room that unfortunately is suffering for that? Wow. I would say also about 80%. Parkinson's impacts movement and the coordination. Two other neurodegenerative diseases, maybe not as common, which is a good thing. Huntington's disease. Anyone in the room know of anyone that is suffering? Okay, a few people. Good. That's a good thing that it's such a low number. Huntington's disease causes motor and cognitive decline. And the final one, I hope I pronounced it correctly, is amyotrophic lateral sclerosis, also known as ALS. Does anyone here know of anyone who is unfortunately suffering with it? Oh, so that number is higher, up to about 50%. Good. So unfortunately, this is something that is not only important for people's research, but also it's affecting people in their everyday lives. It is a great pleasure to welcome our three experts today, and I'm just going to introduce them real quick, and then afterwards we can give them a round of applause. So if you can just put your hands up so that everyone knows who you are. Matthias Jucker, for the German Center for Neurodegenerative Diseases. We have Anastasia Khvorova from the University of Massachusetts Chain Medical School, and Angela Schulz, University Medical Center, Hamburg-Eppendorf. Please welcome our guests today. I didn't have a chance in our preparations to ask, is it okay if we all stay on a first-name basis? I think it's more relaxed for our discussion, even though it's a very serious topic. Excellent. Then I'll be David, of course, as well. Before we begin, someone just came and told me a few minutes ago that we have a special prize winner in our panel as well. I wanted to surprise you with that. Could we give a round of applause to Anastasia Khvorova for winning her prize, the Elsa Krone Fresenius Prize for Medical Research? Congratulations to you. And you can certainly talk about that in a moment, but I just wanted to confirm you were awarded several $1 million for your research. Is that correct?
Correct.
Fantastic. Then congratulations on that. And yeah, I'm sure it's going to be a very prestigious award with a lot of work following it. So, when we talk about neurodegeneration, just real quick for those that are not that familiar with the subject, a few things. We're talking about common pathologies. That means pathological protein aggregation for one, structural and functional abnormalities, disrupted synaptic connections, DNA damage, just to name a few of these pathologies. And again, as I said in this discussion, since some of the people in the room are not that familiar, we're going to try to make sure we explain everything. So, Anastasia, I wanted to start with you. If you could just take us back to Huntington's disease and maybe tell us a few words about what is Huntington's disease.
So Huntington disease is a fascinating disorder because it's been caused by expansion of the repeat in a gene called Huntington. And a lot of neurodegenerative disorders are actually expansion disorders. There's over 40 of it. And what is very unique about the Huntington is that you are born healthy, you live healthy. Actually, there is data indicating that people carriers of Huntington have an average high performance ability. So as a result of it, they're famous journalists, writers, scientists, and then around 40, 50 years, you start to see some neuropsychiatric disorders, depression, anger issues, and then it's what is called career, very typical manifestation, and within 10 years, it's a quick decline to essentially comatose stage, and patients die. It's a devastating disorder because by the time people develop it, they have families, they have careers, and their kids know that they have 50% chance of inheriting, and they're watching their parents die.
Okay, and with this disease, is it generational? Can we say that it's passed on from generation to generation?
It's genetically defined, correct.
Okay, so lifestyle...
50% chance of getting a disease if your parent had it.
Wow, okay. Lifestyles does not play much of a role then in these diseases.
Yes and no, and there is a lot of studies right now in neurodegenerative disorders in general that how active your brain is and how good is your metabolic health has impact on rate of neurodegeneration, and as such, Huntington is affected as well.
Okay, thank you. Question to our technical team, do I have to keep using the hand microphone? This doesn't work? Okay, so we'll keep doing that, that's fine. Mathias, I had a question for you regarding molecular mechanisms and early detection. Your work focuses on understanding the molecular mechanisms underlying Alzheimer's disease and investigating how abnormal protein aggregations such as amyloid plaques and tau tangles, we talked about that the other day, TAU, tau tangles, contribute to disease progression. Could you explain that? And again, for some people who are not in the medical community in terms that we all can understand.
Yeah, I mean, let me tell you.
And please, nice and loud, because there's a door sometimes
Let me tell you why
open.
it is so timely to be here and talk about Alzheimer's disease and, as you said, protein aggregation. I mean, what we found out, and as a community, is that this protein aggregation occurs in our brain at least 20 years before we have any symptoms. That's something we just learned over the last, by the way, decade. And the second thing is we can detect it with very high technical instruments. But what also comes out in these days is we can do it also in blood. So that's the next advantage what we have right now. And the third one is, and we talked about it 2 days ago, we have a first therapy, we have antibodies, and they are not against the symptoms of Alzheimer's disease. They go into the cascade of the pathological cascade, which leads to Alzheimer's disease. They take away these protein aggregates. And that's unique. And to say, to the criticism or to the critics of these therapies or these antibodies that they do only a little bit for the patients, we are so excited because our field thinks the earlier the better. And since we now know it all starts in our brain 20 years before we have symptoms, that's where we want to go. We want to go early.
So just to understand you correctly, we're talking about blood testing to say if you have Alzheimer's or not. Is that out on the market yet? Is it being researched? Clear text?
No, that not yet there, but it's clearly that's where the field goes to.
And we may have like the corona tests in the future where you can go to a pharmacy, pick up a home lab kit and do the test at home. Is that the end goal?
I think most of my colleagues, including myself, would say yes, that's coming.
Fantastic. Well, very good. Okay. I also have a follow-up question for you. You work at the German Center for Neurodegenerative, and I've practiced this word many times, but it's a tough one. Neurodegenerative diseases, Germany's leading research institution dedicated to studying neurodegenerative diseases. Much of your work centers on early Alzheimer's, which is key to halting Alzheimer's and potentially curing it. What are the biggest hurdles, in your opinion, for early detection? Because it is a race against the clock.
Well, the problem is actually with the early detection, we are pretty good. This is not the problem. I think the problem is that, as I said, the techniques to do the early detection are very expensive and can only be done at specialized centers. But the blood, as we just talked about it, that would be a breakthrough. We could do it much earlier. The next hurdle is, why do you want to know? Now we can do it, but why do you want to know if there is no medication for medication there, because right now what is on the market is only for symptomatic patients. So the hurdle would be to do clinical studies which clearly show an antibody or a medication is doing the job much earlier.
Okay. Angela, you're dealing with early diagnosis, especially with childhood dementia. Your research aims to identify biomarkers for early detection, even before symptoms appear. I understand you're also studying degenerative brain diseases in children, especially rare forms of childhood dementia. Could you tell us more about your research and also your findings?
Well, childhood dementia is kind of similar like adult dementia. So we have children that are born completely healthy, and then they go to school, they're very bright, everything is normal, and then all of a sudden they have trouble finding certain words. They also have motor problems, start to stumble, fall more frequently, get cognitive problems, and this progresses very, very rapidly. We have a whole group of diseases that can cause those. Most of them are lysosomal storage diseases.
Could you just speak a little bit louder so I can hear you clearly?
Yeah, we have
Please.
a whole group of diseases that can cause childhood dementia. These are all genetic diseases, so they are inherited. So we sometimes have several children being affected by this in a family. And really here, the research, I think, is so important because in these children, we can see the progression of the dementia and what's happening in the brain in a very pure fashion. These progressing diseases are not confounded by any other comorbidities. These children just have neurodegenerative diseases. And of course, for these children, we aim for early diagnosis, and the earliest would be diagnosing them prenatally, which we can do if we know whether this genetic defect runs in a family, or if we don't know that, which is usually the case because it's recessively inherited, so parents cannot know that they are carrying such a genetic defect. They are completely healthy, all the ancestors have been healthy. This is just a coincidence that two parents meet each other that are carriers of such a disease. That would be newborn screening. So as pediatricians, we are able to screen for about 20 metabolic diseases already. When children are born, they're all tested for that in Germany. And we are now we've just started a pilot project here in Germany, in Hamburg, for testing one of these childhood dementia diseases in newborn screening, where we do have an approved treatment, which is very exciting and encouraging.
So you advocate very much for newborn screening. How could you see that being implemented? Should the Krankenkasse, the Krankenkasse of English, help me out here.
Health insurance.
Insurance companies. I've been living in Germany too long. The insurance companies, that they should cover that? I mean, should it be mandatory? Should it be highly recommended? How should the insurance companies take care of this?
So for the 20 diseases that are already in the regular newborn screening program in Germany, this is covered by the healthcare insurances.
Okay.
Parents are being asked whether they agreed for their child to be screened for these diseases, and then this goes. Now, with the pilot project, we have to prove that the method works and that the early treatment really makes a difference, which it does. We already have clinical trials that show this. And then this is being reviewed by the regulatory authorities, and if the review is positive, then hopefully this will also be implemented in the regular screening.
Excellent. Okay.
Can I just make
Absolutely.
one
Please,
comment?
please.
That one of the things which is being discussed in community quite a bit, that with the development of novel technologies like genome sequencing, with genetically defined disorders, it's not only childhood disorders, you can develop one screen at a time, or you can just sequence a genome. Because if you sequence genome as a whole, it's effectively screening for all genetic disorders, not only childhood. Of course, it has a lot of, and probably cost of that right now in research settings, it's less than $100 per genome. Not in clinical settings, but it's coming. But of course, it has a lot of kind of ethical implications and information sharing, information storage. But it's where field is potentially will evolve.
Okay, excellent. So a question out to the panel in general. What do you think are the current challenges in diagnosing and treating neurodegenerative diseases, especially at the early stages? This is an open question to anyone.
Well, I mean, it's totally different. They are talking about genetically, right, autosomal dominant or whatever genetic causes of the disease, and I'm more standing for these age-related neurodegenerative diseases, where genetic also plays a role, but most of the cases are sporadic. So they go for genetic screening. My approach is totally different.
Yes and no. In Alzheimer, APOE4 and APOE4 homozygous and APOE4 heterozygous in terms of APOE3 allele, it's also the strongest genetic predisposition of the disease progression. And for APOE4 heterozygous, it's 12% of this room. Yeah. So it's not that rare.
Yeah, we just had at the Conference for Childhood Dementia, we had a researcher reporting on studies looking at the prevalence of Alzheimer's disease in heterozygous parents and carriers for childhood dementia. Even though these are different genes, they seem to have a much higher, 5 times higher predisposition to later on develop Alzheimer's. And when we look at pathways affected in the neurodegenerative process, we do see that there might be some joint pathways in the end and some things that we share. So maybe we would find some disease-altering treatments that could be applied to both children and adults.
I think it's misfolded clearance pathways, which is absolutely causative or significantly contributing to Alzheimer, and APOE is mutated, it's got clogged, not mutated, but APOE4, it's get clogged, and in lysosomal storage disorders, it's a confounding factor.
Anastasia, you've done a lot of work with RNA-based therapies, correct? And on my research, I saw that you focus on oligonucleotide RNA-based therapies, which
Good job.
represent... Say again?
You said it right.
Okay. I try my best on these terms. Therapies which represent a major paradigm shift. Instead of targeting proteins with drugs, these therapies target mRNAs, RNAs, for those that don't know, being the molecules that carry genetic instructions from DNA and tell the cells which proteins to make. Can you tell us a little bit more about your research?
So practically, I'm a chemical biologist which got involved in neurodegenerative disorders, not necessarily willingly, but right now half of my lab is working on Huntington, ALS, Alzheimer's, brain aging. And the reason behind it is that while technology has evolved to modulate any target you want, for example, you can modulate APP, which is precursor of amyloid, or you can modulate tau, or you can modulate APOE, where you can modulate JAK1, which blocks inflammation, and clearly this is producing an opportunity to potentially make a disease-modifying drugs. And for Parkinson, it's alpha-synuclein. And a lot of those things are currently either progressing towards clinical trial, or for tau and alpha-synuclein, it's actually in clinical, and APP is in clinical trials now.
How long do you think those trials will take before you come to conclusions? 2 years, 3 years, or it's hard to really guess that?
In neurodegenerative disorders like in genetically defined disorders, in those kids, we now have, actually for lysosomal storage disorders, there is antisense oligo, which is splice switching. And I just last week have seen data from ongoing clinical trial. Within 2 years, you start to see a certain benefit. In Alzheimer's, unfortunately, because it's such a diverse patient population and there is a lot of variation, and also safety is super important. This will take much longer.
Super. Just to our audience, in a few minutes, we'll be opening up the floor to your questions. I'm looking forward to that. I have 2 or 3 more prepared. Preparing for our talk today, I came across CRISPR, C-R-I-S-P-R. So this is gene editing technology that allows for precise modifications of DNA within living organisms, enabling researchers to target specific genes for deletion, insertion, or alteration. Can anyone tell us maybe a few words? It's a very new and emerging gene editing technology, but CRISPR and how it can be used for fighting neurodegenerative diseases.
Okay. Yes, absolutely. And I think I'm a personal believer that in the future it's going to be the case. As of today, in 2025, there is no working technology which gives you delivery of CRISPR throughout the brain. It just doesn't exist. With that said, there is a lot of things which have worked in animals, and it probably will be progressing towards clinic soon. The second issue with CRISPR is it's reversible. So if you do it, you cannot come back. So I personally believe that with oligonucleotide therapeutics, which are reversible, what I'm doing, I'm paving a path for demonstrating safety and creating safety data for CRISPR-based therapeutics, because if you make with a reversible drug, for example, you silence tau, and maybe you see a profound changes, and you show that for 3-4 years or 5 years is completely safe and people are not losing mental acuity, then you follow up with a CRISPR and edited it out. So it's a future, but not probably today.
Okay. Anyone else on that one? Please.
Well, there has just been reports, and I think this is very, very recent, only a few months ago, that one of my pediatric colleagues, Professor Arns Niklas from Children's Hospital in Philadelphia, has, I think, at least for the moment, quite successfully treated a child, a newborn, with a urea cycle defect, which is a disease that affects the liver, with CRISPR, so with gene editing treatment, because that child had a mutation that was applicable for this kind of treatment, and they used nanospheres in order to get the CRISPR into the liver cells. So this has proven to work quite well. The child's doing well. This seems to be safe as for now. This is only a few months. But of course, we now have the challenge to get something like that to the brain, which is the next area. But I think for us as pediatricians, this has really been a breakthrough to really see a child being treated with something like that.
There are clinical data. There is one approved drug for CRISPR, but it's ex vivo. But there is a fantastic clinical data in liver where, like with TTR amyloidosis and several other examples. So probably liver is going to be first. And
could you repeat that? The name of that treatment? You said ex river?
Ex vivo, meaning
Ex vivo,
they take cells, modify cells, and then infuse this back. It's
Thank
a company, CRISPR Therapeutics.
Okay, excellent. Thank you. I've gone to several other sessions too during the Falling Walls, and a big issue seems to be how can we get the academic community and the corporate community together? Maybe in your own experiences, is that going well? Is it challenging? How can we combine the two? Because there seems to be certainly a divide there. Maybe I'll start with you.
Well, I can give you an example. Definitely. So in Alzheimer's disease, there are also mutations which lead 100% or 99% to the disease. And you can predict, you can say if you have the mutation, you get the disease by 55. So in this population, you can do treatments 20 years earlier because you know they're going to get Alzheimer's disease. But these patients are very, very rare. So first of all, there is not a single country who could do these trials. So we do it all together, all over the world. And there is not a single university who can pay for it. So the companies come in and bring the compounds into this population. So clearly this DZNE study, we call it DZNE, is something which could never be done by university academic alone. It could never be done by industry alone, and it could never be done in a single country.
I will say that it's not perfect, but as usually democracy works. It's not working perfectly, but it's working better as in any other form of governance. And for example, in the field of oligonucleotide therapeutics, we organize a society called Oligonucleotide Therapeutic Society, which brings together industry and academic and regulatory and manufacturers once a year in a large meeting which goes in different places in the world. And having this community and a communication, it's super important and really transformative in building the relationships.
One of the big things, microphone. One of the big things is making innovation accessible to the general public. So I wanted to ask all of y'all, how can diagnostic and therapeutic breakthroughs be rolled out in a way that ensures public accessibility, especially considering the high cost and complex administration often associated with such novel therapies? Just an opinion of yours. Maybe I'll start with you, Angela.
Well, maybe an example is the newborn screening that we've just started for one of these diseases, even though technically we could screen for many more diseases and the genetic newborn screening is on the horizon, but for historic reasons in Germany, we're only allowed to screen for diseases where we do have a treatment and where this treatment would really change the trajectory of the disease or even better, prevent the onset of the disease. And this test, it's an enzyme test for this one disease, costs like €5. Very easy, very cheap. And if we start treating the children at a newborn age or before the age of 1 year, we do see in our patients that they do not develop any neurodegenerative disease. And now the oldest ones, this has just been approved, the treatment, are now 8, 9, 10 years old. Normal cognitive development, they go to school, they are bright, they ride a bicycle, they are bilingual, really, really fantastic children, whereas their older siblings would be in a wheelchair, might have died already with a very, very shortened life expectancy. And if we look at that from an economic perspective, even though, I mean, these are children's lives, but also it's €5 against like a 10, 15 years long devastating disease with many, many health costs associated. So I think this treatment might not be completely for free, but it might be expensive, but there's no comparison to the healthcare costs related if a child would become ill.
I will
Yeah,
say that in Europe, you are maybe in a better shape because of the single-payer system. In US, it's even more complicated because of the insurance issues. And while it would be no question that if you look at the healthcare cost for a life of the patient, that preventing the kid becoming disabled from neurodegenerative disorders, overall cost of taking care of this kid, you are saving an enormous amount of money. But you can imagine a situation where insurances are profitable entity which is operating by year. Yeah. So what they care about is a cost of this particular year, and specifically for gene therapy treatments, some of them is a couple of $1 million. For nusimersen, which was disease-modifying small RNA drug, I think first year it's 700,000. So of course that is kind of a discussion to have, and policy making will be transformative in making those drugs available.
And of course, when we talk about insurance, looking at both sides of the Atlantic, here in Germany, it is law if you're working, you have to have health insurance. And in the United States, I think the latest figures is over 20 million people do not have health insurance. So that will also, costs will pay a big...
Vast majority do.
Yeah, the vast majority
do have insurance.
Do have it, true. But still, if I'm not mistaken, over 20 million people do not have it. So if we're looking at a population of 360 million, I understand, but it's still, to me, even though I'm an American, 20 million people not having insurance, and those are the ones sometimes that have small accidents that, I don't want to get into another subject, but they're the ones that really have financial issues with that, and down the road that could also be something to look into. For those that was in my other seminar, 1906 was a date we talked about. Does anyone remember why 1906 could be interesting for our discussion today? Alzheimer's, correct. Excellent. So we talk about in the journalism field, how can we get the rote Faden in German? How can we get someone hooked into the story? For those that don't know, Alzheimer's disease was discovered by a German psychiatrist and neurologist, Alois Alzheimer, in 1906. I double-checked. The discovery took place in Frankfurt, Germany. That means next year we'll be celebrating it's a celebration that he discovered this disease 120 years ago. I say this only for the fact that if you're writing academic papers, if you want to talk about Alzheimer's, it's a very good, especially here in Germany, it's a very good opening line to get people maybe interested in learning more about that. Okay, with that, I would like to open up the floor then for questions. I believe we have someone here with a microphone. If anyone has questions, please raise your hand. Take a look around here. Here, we'll start there, please. Nice and loud.
Hi there. Thank you so much for this great panel discussion so far. I'd love to know about prevention strategies, especially if we're able to detect the anatomical changes 20 years in advance of symptom onset. Thank you.
Well, in terms of prevention, in my opinion, there are two things which are becoming very obvious, and that's one is lifestyle factors. It's unbelievable how much we can reduce the risk of, for example, Alzheimer's disease through lifestyle factors. When you ask me how does it work, which one exactly, that's what we don't really know. We know the numbers, but we don't know how it works. That's one thing which I think is very exciting. The other thing is I want to bring up this word escapee. You may have heard about it. That's a big thing in Alzheimer's disease in these weeks. So these are people, patients, they have mutations which leads to Alzheimer's disease. And we can predict plus minus 1 or 2 years when they should get Alzheimer's disease because their parents got Alzheimer's disease. But in these hundreds and hundreds of people which we follow, they are suddenly escapees. They have the mutation, but they don't get Alzheimer's disease as expected with 45. They don't get it with 50. They don't get it with 55. And suddenly we have a good number with all over the world of these escapees. And nobody knows why. Most likely it's genetics, protective factors, and now the entire AD community is going for these escapees.
Anyone else like that?
It's, it's fascinating, and this is what requires collaboration between different fields. It's early detection and figuring out how to get acceptance of it and how it's paid by insurance and others. It's making sure that the technology platform is in a place to essentially modulate any gene you want for extended periods of time in a patient non-invasive way and safe, and then understanding and coming up with a hypothesis for genetic prevention and understanding detailed mechanism of Alzheimer's disease, Parkinson's disease, and then I think it's getting there, but it will take a collaboration between detection, understanding molecular mechanism of disease, and developing advanced types of safe therapeutics. And cumulatively, this will allow us to get to the point where hopefully your prediction that the number of patients living with Alzheimer's disease will skyrocket will be wrong.
That would be excellent if that would be wrong in the future. Absolutely. That's final there.
We have a similar thing, and even in the childhood dementia field, we sometimes have children within a family where they harbor the same mutation and the same gene, and they might even be twins. We had twin studies, and one twin might develop neurodegenerative symptoms at the age of 8, very severe symptoms, including epileptic seizure, loss of speech, loss of motor function, whereas the other sibling doesn't have any symptoms for another 5, 6, 7, 8 years. That's for a pediatrician, that's a very, very long time difference, and that's something we'd like to better understand, and we think there are more modifying factors there, and maybe they are shared across different types of neurodegenerative pathways.
Excellent, thank you. Looking around, do we have other questions here? I want to look to here. Yeah, we have a gentleman here, please. Nice and loud, please.
So thank you for this interesting panel. I'm also with the Center for Neurodegenerative Diseases in Munich. And we research also Alzheimer's disease and a rare lysosomal storage disorder called Niemann-Pick type C. We've heard a lot about protein misfolding and issues that come with that. And that's why we looked at another disease where protein misfolding is a big issue or cause for the phenotype, and that's cystic fibrosis. And this disease one can now treat much better with drugs that actually inhibit this protein misfolding. And so do you think that this could be a perspective for example, lysosomal storage disorders or Alzheimer's disease? Thank you.
Maybe
Please.
I can answer
Yes.
for the lysosomal storage disease. There are already a few clinical trials in the field of mucopolysaccharidosis where this is being tried. This could be okay for certain types of diseases where really maybe a membrane protein is misfolded. We do it in phenylketonuria. There's misfolding, and there are treatments that kind of target this type of misfolding. So I think this is a very exciting path forward. Absolutely.
I don't know, like, and again, it's if you will have 100 people studying Alzheimer's, you will have probably 10 different views. But in Alzheimer's, misfolding, and with early onset Alzheimer, for sure, it's misfolding and misprocessing, but probably it's not misfolding per se, it's disruption of misfolded protein pathway clearance, which results in a delay of the clearance, which results in more modification and more misfolding. So maybe it's not a primary cause of the disease, it's one of the factors.
Yes, please.
Well, I would love to see a compound which inhibits misfolding through different proteins. And I actually started there too, but in a way it did not really work out for me and for the field. So I think inhibitors have to be very specific. What the next level is, and we talk about neurodegeneration here, is a compound which inhibits neurodegeneration, and this would be good for all these diseases. But this is so tough. I don't have big hopes in that.
Do you think that artificial intelligence could help play a role with that research?
I think we don't understand. I mean, we all talk about this protein misfolding, and then we have the disease.
Yeah.
But how the protein misfolding is linked to the death of a neuron, that's very difficult. And I think there are many, many mechanisms out, but it's so diverse. It's very difficult even to come up with a compound which would help all the different
And I
diseases.
think it's an egg-chicken issue.
Yeah.
It's your protein get hyper-modified and misfolded because it's delayed of the clearance, or it's get hyper-modified and misfolded, and then it's not cleared efficiently. And at this point, it's both of the processes happening, and we do not know whether it's causative or not.
That's a big challenge. Okay, thank you. We have time for questions. Sir, please. We have the microphone for the gentleman here. Oh, I apologize. I'll do this. We'll do him first, and then to you. I apologize. I didn't see you.
Thank you.
Go ahead.
Michael Garner. If you can't prevent death, you can perhaps replace the cell. Stem cell therapy is often discussed. I know you're not experts, but at least for some of these diseases, that might be a way forward. What's your expectation developments in that field?
Well, my understanding is, and I'm looking at my colleague from the Hertie Institute and the DZNE, Thomas Gassrius, to Parkinson. I mean, there was some success with cell replacement in Parkinson's disease. In a disease like Alzheimer's disease, I don't really see how cell replacement could play a role because it's everywhere. That's not specific cells. So I think for certain diseases it may work. For other diseases, age-related neurodegenerative diseases, I don't really see how it could work.
There are very interesting work from the lab of Dan Cleveland, and it's partially controversial, but one of the things they reported for is that there is neuronal progenitor cells, and overall number is not very large compared to number of the neurons. And what they did show is that you can reprogram existing neuronal progenitor cells to differentiate more. And you can do it by modulating of certain targets. And I think the one he's fascinated with is PTP1B. Again, it's a controversial subject, but if something like that will pan out, I think it has a higher probability of being disease-modifying if you are stimulating internal brain capacity to generate novel neurons.
Yeah, maybe in the pediatric field, we have I think quite successful stem cell therapy for a leukodystrophy where the white matter of the brain is affected, for metachromatic leukodystrophy, where the stem cells are genetically modified to not harbor the mutation anymore, and then the children undergo a stem cell transplant, and then these stem cells migrate to the brain and become glia cells and are able to provide the enzyme that's missing in this disease. So I think this is really, really exciting. But this is not a replacement of neurons. It's kind of adding cells that provide a factor that's missing in this specific disease. But I think this is very exciting.
Okay, excellent. Sir, please.
Yes. To what degree viral infections could cause neurodegenerative diseases like MS? It has been proposed Epstein-Barr virus, maybe in Parkinson also it was suggested.
Well, yeah. Well, that's also one of the hot topics, at least in Alzheimer's disease. It started with Corona, with COVID, where also reports came out which are not well done until now, that you have more Alzheimer's disease or less Alzheimer's disease, or the misfolded protein spread more throughout the brain or not. And then came the herpes zoster vaccine. You probably heard about that. And the data are coming in again, and it appears to be true. So if you have this vaccination, you apparently have a lower risk of Alzheimer's disease. But nobody knows how it works. And most people think, at least in my community, it's not specific to herpes zoster. It's more like you give the immune system another boost at a certain period in adulthood or even age. But how it works, I wouldn't know, but I think it's going to be a big topic in the future.
I think while exact mechanism of Alzheimer's disease, in spite of 2025, is not fully understood. For many, many years, we were saying it's amyloid. Clearly, amyloid is an early factor, is a first biomarker, but maybe it's not the end of the story. Then it was tau tangles. And in the recent couple of years, multiple papers which is talking that all this accumulation of debris in the brain is causing inflammation, and viruses will cause inflammation. And this inflammation is causing local disruption of blood-brain barrier, which allows immune cells to come in a brain where they're not supposed to be. And these T cells which come to the brain which are not supposed to be are the ones driving tau neurons, which has a lot of tau. That is one of the models discussed, but as you can hear, inflammation being absolutely key, and I think this is non-disputable. And whenever you have a viral infection, you have inflammation. So it's not, while exact mechanism is not understood, it's clear that anything which is causing more inflammation in the brain, which will accelerate neurodegeneration.
Excellent. We have time for one more question. For some reason, this side of the room has more questions. I wanted to make sure. Excellent. Then we're going to make it 2. 1 and 2. Can we have the microphone? Gentleman here. 3. If they're short, we'll get you, I promise. Go ahead.
Yes. 2 questions. Do you think that from a diagnostic perspective, we're still getting it wrong? Like it's like calling Alzheimer's dementia, it's like calling it cancer. There's actually probably, are there like maybe 100 types? And the second type question would be, do you think that the fact that in Down syndrome patients, they almost inevitably get some kind of dementia, do you think that that's comparable to the sporadic or late onset form?
Thank you. Good.
Well, very short. My understanding from your question was second one first. Yeah, Down syndrome patients get Alzheimer's disease, and for me, it's clear Alzheimer's disease, nothing else. The other thing is, in terms of variety of Alzheimer's disease, that it's not one disease, absolutely. And that's, we also talked 2 days ago with AI and everything. I mean, it clearly you can characterize now different population, and we call them all Alzheimer's disease. So definitely this will come, that we have subtypes, and maybe this is also important for treatment.
You know, like an anecdote. So animal model of disease is what we use to trying to study it. There's 2 types of Alzheimer: early onset Alzheimer, which is caused by mutation, and late onset of Alzheimer, which is truly Alzheimer. They are genetically completely different, but everything in preclinical development is being done in animal model of disease, which is based on early onset Alzheimer, which is actually a completely different disease. So your question is super valid, and I think the field it's almost like a drunk looking under the light because we can, not because this is where the problem is.
Excellent. We have a few more minutes. 2 last questions, one and the final one to you, sir. Go ahead.
Thank you so much for the discussion. So as you mentioned, the brain pathology actually starts decades before we have the symptoms, but the current therapies are evaluating the symptom progression. So as we, especially with the sporadic forms, as we move towards like the more precise diagnosis very early on, do you think we need to redefine the disease in a way that we are able to quantify pre-symptomatic improvements caused by those therapies?
Oh, thank you for this question. I mean, you should have just given this talk, that definitely, so I didn't want to come up with that, but I'm already in our research areas, we say no longer Alzheimer's disease, we say A, T, and N. A for amyloid, T for tau's, and N for neurodegeneration. So we cure A, or we cure tau, or we cure neurodegeneration. And that's extremely helpful, and you actually just gave a nice summary why that is helpful. Also for drug development, for regulatory agencies, that's the way to go.
Super. Thank you very much. Final question, sir.
Hello, my name is Leo. I'm from Neurodegeneration Biologic. We are dealing with exosome EVs therapy for CNS indications. We talked about different indication here. I want to go to the development of a drug and trying to find a biomarker and how it's important for the release criteria of the drug that you want to have, if you can elaborate on that.
Anyone?
I will say the whole field is clinical trials are insanely complex, expensive, and take a long time and super inefficient. So the whole field is moving towards what is called informative phase 1, meaning phase 1 with a biomarker, where you're not looking at clinical manifestation of disease, but you are, for example, reducing amyloid in the blood. It's not a clinical endpoint, but it's a biomarker. And I think that would be, the more we can educate regulatory affairs in value of this type of clinical trial designs, the better we will do as a field.
Yeah, no, sorry. The better we understand the disease trajectory, so studies in the adult patients and the pediatric patients, we have huge international databases that we share and that we also provide as data for pharmaceutical companies to use as natural history control data in these trials. Because especially in pediatrics, I mean, any placebo-controlled trial is very, very difficult in a disease that's deadly. So the more we understand the phenotype of the diseases and what is driving what, the better we can see timely results in clinical trials.
Thank you. At the Falling Wall Summit, we have a tradition, a final question to our guests dealing with breaking barriers. So the final question, and we only have time for maybe a few sentences per each panelist. How do you imagine the therapy of the future, and which walls do we need to overcome to get there? And of course, these are very, very large themes and topics that we're talking about. But in general, for a few sentences, maybe I'll start here with Angela and work way over. 2 or 3 sentences. The biggest barrier you see that we have to get rid of.
I think the biggest barrier is early diagnosis in these diseases and to prevent the onset of these diseases. And the next barrier will be to have really very patient-tailored treatments that might be tailored to a certain genetic defect and that target this defect. And I hope that this will be the future. No onset of disease, and if there's a disease, that we can treat it very, very much precisely.
Thank you. Anastasia, please.
My dream is that within a foreseeable future, we have a collection of oligonucleotide therapeutics which can be administered sub-Q once every 6 months and delivered to the brain, and which will prevent diseases like Huntington, ALS, and Alzheimer and Parkinson from a cure. And I think what is also very important to understand is that while we talk about neurodegeneration as a disease, in reality it's a normal aging process. What you see in an aging brain, it's neurodegeneration, which causes a lot of malfunction everywhere. So
Last sentence, please, because of time.
Absolutely. My hope is that while they have to be precise for certain things, for overall brain aging, I think there will be a lot of commonality in treatment for Alzheimer and just everybody taking it to expand their health span.
Excellent. Thank you. And Mathias, the last word.
Well, the biggest barrier to overcome is the greatest risk factor for neurodegeneration, and that's aging. So if we understand aging, and if we can expand the healthy lifespan, we're not going to have neurodegenerative diseases. And I mean, if you look at venture capitalists, at least in the United States, that's where the money goes. And I'm a little bit disappointed of Europe, which is actually not going with that. They always think we want to extend lifespan. No, we don't want to extend lifespan. We want to expand the healthy period of aging.
Excellent. Thank you. Now that my microphone is working, I wanted to once again congratulate Anastasia. I want to make sure I got it correctly here. The Else Krone-Fresenius Foundation Award. That's for you. Congratulations to that. And please, everyone, thank you for your questions. And thank you to our three speakers, Mathias Jucker, Anastasia Khvorova, and Angela Schulz, for a fascinating discussion. Thank you all very much.